From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on September 4th, 2024. Hello, Micronauts, and welcome to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 57th episode of Matters Microbial. Thank you all so much for listening and/or watching and for spreading the good microbial word. It means a lot to me, truly. As I record this podcast, my microbiology course at the University of Puget Sound has begun. As you can see from this slide of my first lecture, complete with Lux Art by Dr. Jennifer Quinn, I am ready to promote me some microbial PR. I handed out lots and lots of microbial swag, as is my habit, as you can see here. And I think it worked, as evident from this video response from my new Micronauts. As I've stated before, I try to promote OMG, overwhelming microbial greatness, in every lecture. Ditto with WTM, what the microbe, as the class and I marvel at the wonders of the microbial world. I like to start the course with an exercise, and I highly recommend it. I state a word, and the students write down a one-word response to it. I turn those responses into word clouds to show what the students are thinking as they enter our course. The first word I used was germ, and here's the word cloud that resulted. The second word was bacteria, and here is the word cloud that resulted. The third word was archaea, and here is the word cloud that resulted. The fourth word was holobiont, and here is the word cloud that resulted. That's for you, Seth Bordenstein. And the last word was CRISPR, and here is the word cloud that resulted. We're going to revisit this exercise on the very last day of the course and see how the students' views may have changed over our exploration. I am going to have great microbial fun this semester, and I hope that the students do as well. I think it's important on this podcast to discuss viruses, and I've been looking for guests who can share their excitement for viruses and their research with them. So it's an honor to introduce Dr. Tracie Delgado of Seattle Pacific University. She will tell us about her journey into the microbial sciences and how she and her undergraduate team explore how herpesviruses replicate and cause disease, and how understanding their interactions with their host cells may create therapeutic approaches. Tracy, welcome to the Quality Quorum for today. It's a great pleasure.
Thank you, Mark. Thank you for having me. I'm really excited to talk about viruses today with you.
Well, it's something that we really want to do here on the podcast, and you're one of the people that agreed to be on there, so thank you. I think a nice place to start is to tell the listeners and viewers a little bit about your path into the microbial sciences.
Yes, definitely. Well, Mark, I tell you, my path was an interesting one. I would say a rather difficult one as well. And so just a little bit of background, because I think it's an important part of my story, is I grew up in East LA. So I was born in East LA, grew up near East LA, and never had any role models, mentors, people who looked like me that were scientists. And so as I navigated this inner city school system, trying to figure out, what I want to do with my life, I did have a couple of moments where I had some amazing teachers who really invested in me and started to spark that curiosity for science. I remember being in science club in elementary school and really enjoying that and starting to pique my interest. I also watched a lot of Magic School Bus. That was one of my favorites, was where, the germ got inside and they explored the body and the immune system's response trying to fight, these bacteria or viruses in the body. I moved on into high school, and that was really where I started to really think about what I wanted to do with my life. I knew I liked science, but to be honest, the educational system I had was pretty broken. Half the people in my high school didn't graduate from high school, and even I would say 1 in 10 honor students went straight to a 4-year college. It was a pretty hard situation, but I had a really great teacher, and she was my AP biology teacher, and she saw my spark for science. I loved learning about science, and especially microbiology was very interesting to me. And she actually spent extra time in the mornings before school tutoring me extra because I was so excited about science and wanting to pass that AP biology test. And she really, and I did pass it, which was amazing because that wasn't pretty common where I was from. And she really guided me into thinking about a career of being a scientist and a PhD, or going on to graduate school one day. So I'm so thankful for her investing in my life. And I ended up getting accepted into my dream school, UCLA, and I was ready to tackle the world, I guess you would say. And I got to UCLA, and originally I was a molecular cell biology major before I became a micro major. I switched eventually, and my first advice that I got was get in a lab right away. If you're interested in being a scientist, get in a lab. And so I found my way into a plant biology lab and did molecular biology research on plant hormones. And it was so fun. And I got to, work in a lab starting week 2 in college. It was amazing. It's like, I definitely took advantage of that opportunity. And there I started to take classes, and, those general biology classes, I learned more about microbiology. And I found that very fascinating. But at the same time, I was actually really interested in cancer too. So I started to have this struggle of, should I be a microbiologist or a cancer researcher? And at that point, I thought, well, as I moved into my junior year, I thought, well, let me try cancer research and see what that's like. And I loved it. I worked on prostate cancer, myeloma cancer, and signaling pathways that are altered to promote its proliferation. But then I took an advanced microbiology class, and that's when I switched my major, and I thought, wow, microbes are amazing. And I know there's sometimes this little war, what's better, bacteria or viruses? They're both cool. I fell in love more with viruses than bacteria, so I'm sorry. But it was one of my passions of just, realizing viruses are so small, but at the same time can cause devastating disease, not alive, yet we have pandemics and, we have brains and they don't, but somehow, we're in this arms race of trying to combat and fight them. And so I knew I needed a microbiology or virology lab experience. And so I did my first virology lab experience was the summer before my senior year of college. I actually was very fortunate to be placed in the lab of Dr. David Knipe at Harvard, who, for the virologists out there, knows he's a pretty amazing person and has done a lot of amazing science. And I got to study herpes viruses, which after I left, I was very proud to say I love herpes viruses in the sense that I love studying them, learning about them. And I was paired with a great postdoc mentor who really invested time, taught me amazing skills. I did my first time courses. It was beautiful. And so as I left that summer internship, I knew I wanted to do microbiology, but I knew I wanted to do viruses specifically. And that really sparked that curiosity in me, leading into applying to graduate school that senior year of college. And I was still doing cancer research, and I thought, well, I definitely want to go into virology, but maybe I can find a lab that studies how viruses cause cancer. Because then I could bring my love of viruses and cancer, merge them together, and be in my perfect happy world. And so thankfully, I got into the University of Washington in Seattle, into their microbiology program, amazing program. And I ended up joining the lab of Dr. Michael Laganoff, who studied how herpes viruses cause cancer. So it was even better because I already had that love and passion for herpes viruses. And it was during that time where I really felt that formation of a scientist really blossoming. I loved going into the lab. I loved understanding how viruses work, but also uncovering new, I guess, paradigms that hadn't been thought about before, which is the alteration of metabolism by viruses, which I'm sure we're going to talk very soon about. And that really sparked this interest in me in understanding, okay, what causes cancer to happen and how may viruses play a role in that. And specifically, the herpes virus I was working on was Kaposi sarcoma herpes virus, which is one of 8 human herpes viruses that causes Kaposi sarcoma in individuals who are immunosuppressed usually, or it's more endemic in Africa as well. And so maybe not as much in America, but we do see it in other parts of the world. And so that really piqued my interest of microbiology forever. And I still continue to do research on how herpes viruses cause cancer through the alteration of cell metabolism.
What a story when you come right down to it. And can I say there are a couple of threads that are especially interesting to our listeners and viewers. And the first has to do with finding a path. So many people that I meet at the different institutions that I've taught at and been part of, they came in knowing what they were going to do. But folks from I want to be very careful how I phrase this because I don't mean any disrespect to my parents or the social class that they came from, but they were lower working class people and they had no idea how science worked. I was fortunate in that a friend of my brother was very interested in science and I got to be his hands and did a lot of experiments in high school. Again, I found high school and middle school people that were very supportive of what I was doing. My parents never understood any of it. And they just thought it was like some weird hobby of mine. And yeah, I guess it is a weird hobby of mine, but it's one that I love. By the way, my PhD work was on a virus of rhizobium, so I'm not so far from you as you might think.
Okay.
Yeah. Although bacteriophages, I find virologists tend to sometimes look a little bit askance at bacteriophages. But it's so interesting that you say this because when you look at a very simple virus like tobacco mosaic virus, it's composed of one strand of nucleic acid, one type of protein. It can crystallize, and yet decades after, you abrade the leaf of a tobacco plant, rub a little bit in it, and it takes over the metabolism of the host because that's what viruses do.
They do.
Right. So I thought what you might do, if you wish, is talk a little bit about herpes viruses because every time I talk about them, I get folks who giggle and it isn't funny, right? Even though, you know, it sounds like a joke from Saturday Night Live, but no, it's not. It's a serious disease, as some people have talked about, and it is relevant and it's a great model system. So could you tell us more?
Yeah. So herpes viruses are a big family. There's actually 3 categories of herpes viruses. There are 3 families. There's alpha, beta, and gamma. And so, as you can see on this image, these clusters of families include the alpha family, which have herpes simplex virus, which can cause cold sores, which is herpes simplex 1, herpes simplex 2, which predominantly causes genital herpes. And it also includes varicella zoster virus, which is the one that causes chickenpox. And so those are the main alpha herpes viruses. And then there is the beta herpes viruses, which include the human cytomegalovirus, which can cause birth defects. Unfortunately, women who get pregnant while they get infected with this herpes virus, their baby can have birth defects when they're born, which is very, very sad and unfortunate. And it also includes, when we look at the gamma herpes virus family, Kaposi sarcoma herpes virus, which is one that it's a family, or really the gamma herpes viruses are the ones that cause cancer. And so Kaposi sarcoma herpes virus causes Kaposi sarcoma, which is an endothelial cell cancer. It can also cause B-cell-like cancers or B-cell cancers. And then there's also Epstein-Barr virus, which a lot of people know as mono or causing mononucleosis, but can also cause B-cell lymphomas. And so when you look at kind of, and then there's other herpes viruses in there, like human herpes virus 7 and human herpes virus 6, which cause like a roseola or like rashes. They seem to come once and then disappear. And actually, herpes viruses in general have what's known as a latent versus a lytic life cycle, where the first time you get infected, you know, just like any viral infection, you're sick for about 2 weeks, because it takes about that long for your immune system to fight back, make antibodies from those B cells. And then as soon as your body fights back and that immune system response happens, they go what's known as latent, which they'll hide in some sort of host cell. Sometimes their DNA will be tethered by a protein on your host genome. Sometimes they may make non-coding RNAs that may stick around as a genome copy. And so there's a couple different ways in which it can go latent. Sometimes it's a completely different cell type than the original infection. So for example, when you get a cold sore, that viral productive stage called lytic infection is it replicating in your epithelial cells of your lip. And then your immune system fights back and it runs into your trigeminal ganglion, which are neurons, and it gets back in here and it hides. So then it's pretty much a waiting game. Will your body be able to fight that virus forever and keep it at bay, keep it latent? Or will one day it reactivate, which is pops back up into lytic replication, producing new virion or viral particles? And when it does so, and interesting with HSV1, which causes cold sores, is it'll actually go back down that nerve, straight back to the original site you originally had it, and replicate again.
Isn't that
now what caused?
I was going to say, isn't that fascinating that it has a path and it follows that path later? And it's apparently, from what I've read, it's co-opting machinery inside the cell to move it from place to place, which is to me fascinating. And I love telling students, have you ever had a cold sore? I'm very careful. I don't say, have you ever had herpes? Because that, as I say, and then I say, it's hiding out right here. And it's so strange to think about, isn't it?
It very, very much is. Yes. And then the question is, well, what allowed it to come back out again, whether it's herpes simplex 1 or whether it be Kaposi-sarcoma herpes virus, right, which could cause cancer. What is from that initial infection or that initial fever response that you have, that virus goes latent, what's making it come back out again? What allows it to go what's known as reactivate? Mm-hmm. And so a lot of it has to do with our immune response. It may be weakened due to multiple reasons. It may be due to another illness. It may be due to immunosuppression or drugs you may be taking. It could sometimes, even for certain viruses like herpes simplex viruses, they've been seen to reactivate due to stress or even hormonal changes or menstrual cycle changes. UV light. There's lots of research out there trying to figure out what's allowing this to come back out and replicate lytically. And that lytic productive replication is how the virus is able to spread, right? Once it pops back out, that productive infection makes you infectious. And now that virus can move on. Or in the case of a person who may have a virus that can cause cancer, it can seed new cells with virus, which can one day become cancer or tumors. And so in general, herpes viruses I find are really fascinating. And I've spent time throughout my scientific career studying both the latent side of viral replication and its alteration of metabolism. And now more recently, I've switched into the lytic replication of and how it alters metabolism.
So may I jump in for just a moment?
Yeah.
So one of the things that I like to talk about with students is this strange idea called cellular microbiology. And it doesn't mean the cell biology of microbes. It means how microbes interact with our cells and how that process of interaction has taught us a lot about how our cells work. For example, organisms like Listeria, they are able to inside the cell co-opt the cytoskeleton to make what are called actin rockets to push them into other cells. So we know a lot about how actin works as a result of this. So in a similar way, when you think about viruses in general, they're completely reliant on the innate metabolism. And so they are able to flip switches inside the cell to turn it into a factory for making more virus, is one way of looking at it. And when they're latent, they have another set of switches that they pull, which makes viruses not just interesting in their own right, but of also interest to people who are fascinated by how our own cells work. So what I'm doing is congratulating you on your work.
Well, thank you very much. Yes, I think that when so thinking about those switches, as you just mentioned, I think that's a really good point, and something we should think about is, you know, as a cell that is latent infected, right, or latently infected, silent virus sitting, mainly the cell's not dividing much at that point, right, depending on the cell type we're talking about. That virus wants that host cell to survive. So it's going to flip these switches that are going to promote cell survival, evasion of cell death or apoptosis, right? And inhibiting any immune response that may be able to recognize it. That's going to be part of that survival. But when you think about that, it also can be one of the hallmarks of cancer. And so not all viruses cause cancer. I think when you have particular viruses that may not only cause immortality or cell survival because of latency, but also may have other types of genes or other things that encodes to also increase cell division or other things later, all taken together, this is a really interesting thing. But at the same time, then you move on to a lytic active production replication. And those cell metabolism changes have to be probably a little different because at this point, you're building brand new viruses. That cell is going to die in about 24 to 48 hours, depending on which herpes virus we're talking about. And so it's not as much concerned about cell survival. It's concerned on its replication and how do I flip those switches on this cell to make more proteins, more nucleic acids, more energy, ATP, more lipids, if it's a lipid envelope virus especially, to promote its self-replication. And so I think that actually brings in, if there's a slide which shows just in general how viruses replicate. And as we just mentioned, right, viruses are these intracellular parasites, and they completely rely on that host to promote more copies of themselves. And so when we think about that, those copies, as I mentioned, will be amino acids, nucleic acids, lipids, ATP, multiple other precursors to replicate. And again, a virus is not alive, yet somehow it absolutely requires the metabolism of something that is alive, our own cells, and takes over and changes the landscape of that cell genetically by turning on genes with its own viral proteins to express certain genes, to pretty much change the landscape of that cell's metabolism to promote its replication.
Oh, I think it's just so cool to think about. And that means, by the way, can I tell you something I'm kind of sad about?
Yes.
So I used to teach Introduction to Cell and Molecular Biology, the first year biology course, the unity of life. And I no longer do because I'm in phased retirement now. But one of the things I talked about a lot is this transition between regular cells and cancer cells having to do with essentially flipping switches with cell signaling and certain other processes. And what you just said is something I would have emphasized in my course. So I want to thank you for that. And it's important to remember this idea of anything that lives in our cells kind of being like a puppeteer. And they're making all the modifications to make it a home for themselves. So I think sometimes it's viewed as a martial thing. You're taking over, you're turning them into a factory, but it's more nuanced than that. And I want to thank you for the way that you described it.
Of course. It's interesting you say that too, because a lot of the viruses, when they alter metabolism, they're flipping on the switches of master regulators of metabolism. So when I was actually in graduate school, brought me to this project was a previous graduate student found that Kaposi-sarcoma herpes virus induced the expression or I guess stabilization of a protein called hypoxia-induced factor, called HIF. And traditionally, HIF has been studied very extensively in cancer. Pretty much what happens is that HIF will turn on the expression of many genes, especially those involved in glucose metabolism. And those genes encourage the cell to eat more glucose, to break it down quicker, and to produce energy. And metabolism I like to think about it, even though I know it could be a little scary sometimes, just looking at those huge maps of metabolic pathways. I like to think about it like the US highways, right? We have so many highways here in the United States, and we got some central ones, like on the West Coast, we all know, I-5. I don't really know what's on the East Coast, I'm sorry, but at least on the West Coast. And I know we have some major ones that might go, from west to east and things like that. But they all interconnect at some point, right? If you block a highway, people will flood the other parts of a different highway to eventually get to where they need to go. And so I think about metabolic pathways like that, right? Is that you're basically, when you're increasing the flow or flux, what they call, of a particular highway, that can eventually feed into other parts as well. So maybe that glucose is not just feeding to make ATP, it can also be used to make and shuttled off another highway to make nucleotides, or shuttled off eventually another way to make fats. And so when you think about these metabolic pathways in general, they are really interconnected. And by increasing the master regulator HIF in these KSHV infected cells, they induce HIF expression, hypoxia-induced factor. That really brought us to the question of, well, we're studying a virus that causes cancer, and we know that HIF is really increased in cancer. And this is what really brought us into this whole brand new field when I was in graduate school, is can a virus alter metabolism and cause these cancer-like effects, which HIF is a regulator of. And this was actually coined back in the days, the Warburg effect, and it's still very popular now, where there was a scientist called Otto Warburg who discovered that cancer cells eat sugar faster, and they metabolize it faster, which is going to promote its own replication. And it's actually an interesting phenomenon, because this is how a PET scan works. For anybody who's ever gotten or heard of someone getting a cancer scan, they give them this labeled glucose, and they get it through IV, and any cell that's eating sugar faster is going to glow, and you can see it on that scan. And so that original, that's going to tell you where the cancer is, because you can see these organs lighting up, or parts of the organs lighting up due to that increased sugar metabolism. So in graduate school, we found, a previous student found, hey, KSHV infected cells increase HIF. And then he said, let's check and see if we see this Warburg-like effect, which is an increase in glucose uptake and metabolism. So he started these preliminary experiments where he saw that, yes, if you feed KSH2 infected cells, they consume glucose faster than non-infected cells. And that led to my project, which is I joined the lab right at that time, and he was graduating, and he said, hey, this is really interesting. You should follow up on this. I think there may be an alteration in metabolism. And so that's where my project started. And from there, I was able to continue investigating and see not only do you increase glucose uptake, but you increase lactic acid production, which is also part of the Warburg effect. And really interestingly is that when cells produce increased acid, that micro pH environment makes it harder for the immune system to react against that cell, because you're basically putting this little buffered space around the cells. And not only that, it's also interesting that cells that undergo the Warburg effect, which I also found in KSHV infected cells, they not only increase glucose uptake to lactic acid, but they slow down mitochondrial oxidative phosphorylation, which is the use of oxygen or the reliance of oxygen. And you got to think about it this way. When a cell is going through glucose metabolism, if it's a virus infected cell, for example, you want to make energy very fast. And because you only got a certain amount of time to make virus before that immune system comes and gets you, right? Those immune cells. And so you got, again, they don't got a brain, but there's something about viruses, right? Glycolysis, you can ramp that up 100 times faster and make more ATP by just increasing glycolysis, assuming unlimited substrates and able to bring in that glucose, than by going through oxidative phosphorylation. And interestingly, oxidative phosphorylation, if you finish it all the way through the electron transport chain back in the days of Gen Bio, when you learn that, it increases reactive oxygen species, which are very dangerous to the cell and can kill it. And so it's actually more beneficial to just do glycolysis or glucose, breakdown to lactic acid very, very fast than to go all the way through where you would originally usually get more yield of ATP through oxidative phosphorylation, only because assuming you can bring in that glucose faster and break it down faster, you make more ATP that way and in a safer environment than if you go through oxidative phosphorylation.
You have no idea how happy this is making me, because I've taught about this issue of glycolysis versus fermentation versus oxidative phosphorylation for many years. And, see, it ties all together. This is what I was saying before. You can think of this as being a kind of an academic way of looking at things, but it's also something that can help with the detection of cancer, as well as understanding how viruses work. So this is what was hard for me in biochemistry when I was an undergrad. And then when I became a graduate student, I understood that the interconnections made things clearer to me.
And so thinking about that concept, thinking about, okay, how are they, what is this connecting to, right? They're at the time when I was first studying KSHB and its alteration of metabolism by a virus, like that was a very new field. There was just a handful of publications just starting to investigate, viruses and their alteration of metabolism. And I was coming at it from the view of like, this is a virus that causes cancer. Alteration of metabolism makes sense. It's a hallmark of cancer. So, but at that point, I wasn't really thinking, well, maybe all viruses do this, not just the ones that cause cancer, but it is a requirement as one of the hallmarks of cancer. There's other things that the virus, there in my lab, people were working on how the virus induces angiogenesis, which is the creation of new blood vessels to feed the cells, right? Or other ones looking at other signaling pathways or evasion of apoptosis. So there's obviously many facets that lead to cancer. But at that point, it wasn't very clear, do all viruses alter metabolism? How do they alter metabolism? Is it all the same, or do they pick particular paths or highways that are most important for their life cycle and their production? And so when I was studying, I was realizing, okay, this herpes virus is increasing what's known as the Warburg effect, glucose metabolism, less oxidative phosphorylation, less oxygen consumption. I actually did this by using this machine called a seahorse, which was a really, really cool machine, which has these little tiny wells, and it measures oxygen and pH in each well. So I can take infected cells and non-infected cells and put them through a machine, and the seahorse machine can measure in that well how much oxygen is being consumed and how much the pH is dropping by lactic acid production. And so through that, we were able to see, wow, there is an increase in lactic acid production, and there's a decrease in oxygen consumption, which is the end of that oxidative phosphorylation chain. But then the question is, okay, well, why are they increasing what is the virus doing with this increased glucose? It could be for energy, but it can also be to feed other metabolic pathways. And so the pathways that I studied at that time, and this was all latent KSHB, so this was not lytic. At that time, I was looking to see, well, what other pathways may be induced in cancer that follow this path. And one of them was the fatty acid synthesis pathway. And so, as you probably, or most people know, if you eat a lot of sugar, your body stores that sugar into fat. And I love sugar too. Have to watch those sugar calories sometimes. But what's interesting is that there are particular cells in our body that have activated or have that highway open to create fat. And you probably know where they're found based on where your weight's at. But we have these cells called adipose cells. And those cells are the ones that take that excess sugar and make it into fat. Normal cells, like endothelial cells, don't traditionally have the fatty acid synthesis pathway activated. They have the genetics for it, like any cell has the DNA for everything, but those genes or enzymes are not expressed. And so non-cancerous cells or non-infected cells, normal cells, don't express the fatty acid synthesis pathway. It's like putting a little barricade on that highway saying not in use, right? But these virus-infected cells are able to, which I found as well in graduate school, was that they're able to open up that highway and start turning on the expression of genes that lead to the production of fatty acids, brand new fatty acids, not from the diet, because sometimes cells may just eat more fats to get their fats. No, this is like brand new fats being produced, which the virus is basically manipulating that host to turn on genes to create fats, which, being a lipid enveloped virus, it would make sense because that can be used as well as to make lipids as it builds its virus and exits.
And so how does all of this reflect the research that you're doing with your they are your students, but they're also your colleagues. And I think that's something that we talk about a lot. It's a group effort. It's not hierarchical.
Yes. Yeah. So that was the end of my PhD studies. And I knew I really wanted to continue studying how viruses alter metabolism. And so I moved on into thinking about, I was really, really jealous of all the people who could do plaque assays, to be honest, and do lytic replication studies. KSHP, as well as EBV, Epstein-Barr virus, it's really difficult to do plaque assays, which is a way to measure virus production. And so it just doesn't naturally go lytic in culture the way that another gamma herpes virus, which is in that same family known as murine herpes virus 68. So I knew I was going to go and move institutions, and I wanted an undergraduate lab, and I wanted something that I thought was very relevant, that was safe, that would go lytic and replicate very well and can form plaques. And so since pretty much at that point, and even until, I was able to publish it, no one had studied the gamma herpes virus's changes in cell metabolism during lytic production. And so the whole thing was on latent, that silent infection. So I wanted to see, okay, what's happening during that productive infection when the virus is replicating and making new virus particles? And so I switched, as you can see again in the slide, to this viral model system known as MHV68 or murine herpes virus 68, also called murine herpes 4. And this viral system is fantastic. It can infect multiple types of cell lines in vitro, including mouse fibroblasts, which is what I use. Traditionally, in a mouse, when it first gets infected, it replicates in the lungs for about 2 weeks until that immune system fights back. And then it goes latent in B cells in the spleen. And one day it can produce B cell lymphomas if it if it progresses to cancer. And so, but I wanted to do everything in vitro. And so I decided, let's go ahead and metabolically profile what is occurring during the lytic cycle of gamma herpes viruses, that family of gamma herpes viruses. So using MHV68 as a model system, we were able to show that when this virus infects cells, we were able, and this was fun for the students, I have to say, taking their first time points all through the days and nights for multiple rounds. I think that's when you realize you're really a virologist, is those time points, right? And so we were able to metabolically profile all of the changes in metabolism occurring throughout multiple stages of that replication cycle for up to 24 hours, about every 4 hours or so. And we found, interestingly, that there was an increase in glucose metabolism. There was also an increase in glutamine metabolism, which was something that I started to look at in collaboration before in KSHP, but it was something that I was really interested in because what we found, and you can see in this image here, is that when you starve cells of glutamine, these infected cells, the virus can make like almost no virus, like 2 plaques at no dilution. Like it was pretty much obliterating the virus production. And so this was very interesting to us to think that, by starving of glucose, it did decrease virus production, but not as much as glutamine.
Interesting.
And glutamine is another carbon source that can not only be used to replenish, say, the Krebs cycle, the TCA cycle, and broken down. It can be deaminated and use some of those amino groups for nucleotide production, but also combined with other amino acids, it can also fuel nucleotide metabolism in general to create brand new nucleotides. And so that was really interesting. We also found that the most common and statistically significant metabolites that were increased during infection were all nucleotide, or almost all mostly nucleotide, not all nucleotide, mostly nucleotide. So it showed that, while we're looking at latency in human herpes viruses, it's more fatty acid synthesis and glycolysis. Moving on into the lytic cycle, we're seeing more glutamine metabolism and nucleotide metabolism being more important to produce brand new viral particles. And so from that research, we also started to dabble a little with using metabolic inhibitors as a way to stop virus production, to see if that's a worthwhile endeavor. And so one of the first tests that we tried was not only by starving glucose or glutamine, which is just a carbon source, we also tried 2 different drugs. One of them being 2DG, which is 2-deoxy-D-glucose. It's actually very similar to what they give the people who get PET scans. It's like a glucose analog. So it's like a fake glucose that when you eat it, it can't be broken down any further. So it's kind of like a blockage of your pipe. It doesn't allow the drainage to continue. And so once you feed these cells 2DG, it slows down that first step of glucose and glycolysis metabolism. And so you could see in this image, this 2DG drug, when they were fed to these infected cells, without 2DG, viral production was at 100% capacity. The control is set to 100%. And when you add 2DG, the viral production goes down very dramatically. And so that was really interesting to us. But again, what direction is that going, right? And can 2DG be used more universally? A paper actually came out a couple years ago during the COVID pandemic where they found that this 2DG is actually a FDA-approved drug used in cancer patients. And so they said, let's give it to people with COVID who are on a ventilator because we don't know what else to give people at this time. You may remember that time where people were like, let's find this drug and what's out there that's found to be safe and repurpose it and see if it works. And they found people who took 2DG got off the ventilator twice as fast, 50% of, so they might have been on it for, they could have been on it for 5 or 6 days and they got off of it in 2.5 to 3 days just by giving them this drug that it slows down metabolism. And so thinking about, this in the grand scheme of things, repurposing these metabolic drugs would be great. And thinking about drugs that can block these aspects of metabolism that we know in the virus I work on increases, but we also see other herpes viruses to HIV, to dengue virus, to hepatitis. All these viruses are increasing metabolism. And they may be doing it uniquely. Like some might, do more glutamine metabolism than glucose, or some might do more fatty acid. right? But if we have these networks of drugs that we can say these are FDA approved and they work to inhibit glycolysis, and these are ones that work and inhibit glutamine metabolism, and these are ones that work and approved for other diseases that work against nucleotide or blocking nucleotide metabolism, then we can start having these broad antivirals that can be used and repurposed should we encounter a virus we don't know how to treat at this time, until we can get more specific therapies. And I very much encourage very specific therapies, right? During that time.
So the good news here, when you think about it, is that no matter what type of virus you're talking about, they're modifying and co-opting aspects of the metabolism of the host cell, which means that's a common thing that you can use as potential therapeutics.
Correct.
Wonderful.
Yes.
Another question that's occurred to me as I'm listening to you is, what are the effects on some of these metabolic inhibitors on standard cellular metabolism? I mean, it's always a challenge to find something that targets the virus. But what you're doing here is something that's also potentially going to target normal metabolism.
Yes. And so you do want to, anytime you target metabolism, all cells metabolize at different rates, and different metabolic pathways can be more important than others. So there's usually going to be some sort of side effect as part of that process. So for example, any drugs that inhibit fat metabolism, most cells don't make as much brand new fat. So normally that's not a problem, but it has been shown to cause some weight loss in that process. So weight loss drugs have been used and repurposed as antivirals. There also has been, if you use a nucleotide metabolism inhibitor that prevents the creation of new DNA and RNA, that can affect dividing cells like your immune system cells, for example. So sometimes that can have a side effect of like fatigue and things like that. But I like to think about it in the scheme of, there will probably be side effects, but if this is a virus, you want to hit it early. You want to get it as early as possible before the virus has a chance to reach that as it's exponential, and it's, so the sooner you can target with the drug, the faster it is to bring virus levels low enough that the immune system can tackle it instead. Because once it goes too high, it makes it harder for that initial innate immune response to kind of take over. And you don't need to take it that long, because you eventually will make B cells with antibodies. So you will, about 2 weeks is the maximum of how long you would need to take these drugs. And these drugs that I'm looking at are already in the market for cancer, rheumatoid arthritis, other types of diseases. And so they are already being used for long terms, months on end. And so there definitely is important to think about, which metabolic pathways will they harm for normal cells, or at least all cells metabolize, what cells can be part of that, have as a consequence, a side effect, not good side effect. But in the long term, you won't take it for long enough where I can see it being an issue. However, as you saw with the COVID pandemic, you could have people dying if there's a pandemic virus. So if you can save someone's life in those 2 weeks and maybe feel a little fatigued, I think it's worth it compared to, what the other option could be as the outcome of a virus.
And I think you alluded to this, but clearly the timing of the treatment is important. Earlier is better.
Yes. And I think that's true of most virus antivirals, right? We want the earlier you can give the antiviral, that's when the virus is just starting to climb. You're able to, even though it already reached this high, you're stopping new replication. And that gives the immune system a chance to get rid of whatever virus is already there. And of course, the further along it is, the harder it is, but it's not to say it's hopeless, but it's just in general that you want to catch it as early as possible during those initial first symptoms. And so thinking about the methods of, testing these kinds of drugs, as you can see, when we first try and identify a drug that we want to test, the first thing we want to make sure is it's safe. Right? It's FDA approved, but we're testing in vitro. So we say, okay, what dose of this drug is safe? So with my students, we look through the literature. Where is it used already therapeutically? What drug concentrations do they use? And we go from there and we do this drug safety curve. And we find doses of drug that are first, they look at the pretty pictures under the microscopes, and they can tell, they learn these cells looking stressed, or they're looking a little like not so good or starting to die. Okay, let's not do that dose. And we just go down and down and down. And they also do cell counts and cell viability counts to see, okay, we want, my goal is that we're not killing more than 5% of the cells, and usually you have about 2 to 3% death already. And, you know, we want to find something that is going to be healthy, or at least be tolerated by normal cells. But the infected cells, I don't really care if they're trying to die or not, because that means it's fine. Like, you know, it's the normal cells. So we do these drug safety curves. Once we find a good dose, as you can see here as well, we go through a different method where we find that safe dose, and we usually try a couple doses. And we take these infected cells and we add no drug, control solvent, or the drug dose we found to be safe. And then they go in and they collect the virus that's produced 48 hours later, which is traditional for our viral system. And from that viral media or supernatant, they then will not only do cell counts, they'll do viral production assays using plaque assays to identify virus production, but they also will just double check and do cell counts and just show how healthy are these cells at this point. We have non-infected cells as well that are set as controls side by side every time. And then once they do those plaque assays, you know, I definitely have gotten a lot of screams and excitement when they find a drug that works. I've even incorporated this into my medical virology class in the classroom, where they screen through drugs and start with qualitative drug dose. You know, they'll look at just microscopic pictures and do plaque assays, and then we refine that in my research lab with my students. And so then through that, we can say, okay, is this drug working? And excitingly, you know, we're really starting to see that, at least in my viral system, or the viral system I work on, MHV68, that nucleotide metabolism seems to be really, really, I guess you would say, a significant portion of the alteration of metabolism in these gamma herpes virus infected cells. And so you'll see another photo there that shows this drug, which we call drug A, which is a drug that we just got out a couple of months ago. So it's preliminary, but it has been replicated many, many times. And it is a nucleotide inhibitor drug, which is out and FDA approved already. And we're able to show that this viral production drops dramatically to basically, again, almost zero. And going back to that glutamine starvation dropping virus production, we believe that glutamine is actually being fed into the nucleotide production. And we also believe that maybe as well, the glucose is also being fed into nucleotide. But we have to, of course, continue to test those hypotheses and continue working with that. But we have some good preliminary data that is supporting those aspects. So we are really excited to continue screening through probably about 10 different FDA-approved drugs that are metabolic inhibitors, and seeing which ones work and which ones don't, and which can be used to maybe help with gamma herpes virus infections. But also, these drugs could hopefully be a starting point for others who are studying other viruses, and they can see, will this work out great for, you know, as a good broad antiviral for their virus that they're working on.
You know, I hear all this, and my knee-jerk reaction is to say, I want to come work for you. And it
is a fun lab, I have to say. I love the projects. Yeah.
How exciting for them as well. And you have undergraduates. Is that all that you have in your laboratory?
Yeah. Mm-hmm. So I'm at a primary undergraduate research institution. I have no postdocs, no postbacs, no PhD, no graduate student, nothing. Like, it's all undergraduates, and it is amazing. I love mentorship. And I am a firm believer that undergraduates can do advanced science given the opportunity and the right mentorship and skills and training. And so the students in my lab are the ones who do all of the experiments. They're running around all the time and really enjoying it, as I could see as well. We have in our university as well, like I mentioned, I have that medical virology class where I just made it into a research-based course, so I can bring that excitement as well into their lab, and they were super excited. I have to say, to give them a research project, give them a drug and say, go search the literature and start finding doses and try out for the first time. Like, I think it's one of the best tools to form and encourage new scientists to enter the field.
Oh, I can't agree more. I mean, I'm at a PUI myself, and I don't do like this fabulous stuff that anyone can look at and say, hey, I can see the applications of this. But what I do think is that you've really hit the nail on the head, metaphorically speaking, by the concept of ownership. I find, certainly with undergraduate students, the sense of a project that they're responsible for, they get the positive strokes from it. You and I both know that when they come to you with a result, their eyes shine. And not always the ones you expect. Like, I've had people that are way too cool for school, and they completely nerd out when it's something that they found. And there's no substitute for it. When I was an undergrad, I worked with postdocs and graduate students, and I did my part of it, and that was wonderful, and I'm very grateful for it. But there's no substitute for something that's theirs. And what I love about your work is it's super relevant to health issues, it's super relevant to basic cell biology, and it's super relevant to something that undergraduates can test.
Definitely. And if I can give a couple of tips too with working with undergraduates, I think especially thinking about inclusive mentorship, I think about my own background. I think about, needing mentors and people, to help me catch up academically. I think a lot of the times you see students and they're capable, they just don't have that background that if you give them certain resources, they can catch up and do amazing and even do better than you would have ever thought they would have. And so every student that comes into my lab, the first thing that I do is I give them a handbook that includes, so I think it's like important to have this like knowledge-based mentorship to start where, some person might have heard of cell media or cell culture and other people may not have as much in their background, right? And so it contains experimental principles, everything from like, what does serum do, or how does trypsin work, and how do you inactivate it, in a culture, or so you're basically filling in any gaps that they might have in knowledge, and it really helps with troubleshooting. So you have students who may have come in with more knowledge and ones that don't, but you're giving these students the opportunity to learn and catch up. And with that motivation that I've seen with these students, they are motivated to learn and catch up. Every student also, as they usually come in as juniors, but sometimes come in as sophomores, they write an NSF GFRP graduate proposal. From the first, like, that's one of the first meetings we have when they come in. So that really makes them think, what am I actually doing? Because I've been in labs before where I didn't really know what I was doing all the time because I didn't, if I would have had that mentorship saying, okay, this is the background and this is why, and what are the aims, and go find papers and guide you to read literature. To write an NSF style graduate proposal is a pretty big deal, especially when you only got those 2 pages, right? You got to like be concrete, as you know, right? And so they write that proposal, but it really gets their brain thinking and get excited. And then, of course, there's, teaching them experimental techniques. But I find words of affirmation are really important. You really need to, encourage them on the things that they're doing right. And really think about the words you're using when you're speaking with your students. You don't want them to have trauma or think that they're not cut out for the field. Giving them those words of affirmation to say, when mistakes happen, instead of harping on, oh, that was a mistake. Instead, it should be, okay, that's interesting. So what did we learn through that? Let's think about, some backgrounds and principles and why the mistake led to this outcome, just to start get those gears turning. And then, but at the same time saying it is, it's okay. That's part of growing and being a scientist. Just like when you're learning to ride a bike, you fall a bunch of times and, you get back up again and you keep going. Right. So what did you learn when you fell? And now let's continue working through that.
It's not a mistake. It's a lesson. And,
yes, for sure.
And there's also this concept that I learned a long time ago from one of my mentors about the in research, that in order for something to be true, it has to be replicated by somebody else. And you have to kind of have that attitude about it. And that really separates things. I really like what you've described here. And I really like that you're creating, and we talk a lot about this term, and if terms get used too often, they lose their power. But we talk about a welcoming environment, and it's used all the time on my campus. And I kind of think it shouldn't be, because I want that term welcoming environment to mean something. I want to have a welcoming and supportive environment for my students, and so do you for yours. And we need to really emphasize that, I think, because you just can't tell. You really don't know about the path that someone's told. Also, we've been kind of infected by what I call the Yelpification of our society, where we have no trouble saying something negative, but we're very sparing in our praise. And I'm not saying false praise. When someone does a dilution series and they're able to show that they're getting tenfold differences, say, when they're doing colony counts, that's a big deal.
It is, for sure.
And you sometimes have to fight the students to say, you know what, this is a big deal. I am proud. It's not fake.
For sure. And giving them those opportunities to be self-independent. I think that's also key. I mean, being at an undergraduate research lab, they are my graduate students, right? They're my students who created all the data for publications, right? They're the ones so they all have their own project, which leads to self-confidence and independence. But also, as they become seniors, I have the seniors mentor the juniors, yes. Which also research shows that when you do that, it helps promote this self-efficacy of I can do it. Because now I'm helping, look at how far I've come. And I sometimes have to remind them, like, trust yourself. Remember, you know more than the ones who are coming in. Even if you don't have all the answers, it's okay. That's part of the journey. And then finally, I think it's important that they're involved in the community. I think research shows that being involved in the scientific community, being around people who look like them, attend conferences, other people from other universities, big and small, and doing science, that shows you're part of that community and leads to that self-efficacy of I am a scientist as well.
We have funny ideas about what it is to be a scientist. And if you'll forgive this joke, there's a thing called a big tent. We need a big petri dish, because we really need different faces and different voices, because it should be for everyone. And I know that sounds all like a Hallmark card, but it's something I genuinely believe.
Yep. And different voices all think about science and the way to answer those questions differently. So diversity in science is very important to start thinking about new ways and new things to study in science.
Oh, Tracy, this has been a simply wonderful episode, and I am so grateful to you for this. I also want to say to listeners and viewers that in the show notes, I'm going to include a link, obviously, to Dr. Delgado's website for the laboratory group that she's part of. And it is so inspiring to read on every different level. There's so much there for everyone. And I hope that every listener and viewer will give it a look. And I want to absolutely, and I want to thank you for your time and wish you and your family and your students / colleagues just the very best. Thank you for your time today.
Thank you for having me, Mark. It was such a pleasure to talk with you today.
I have to come up and visit.
Definitely.
Thanks again. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode with, as I mentioned, absolutely essential links can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Tracie Delgado is in the biology department at Seattle Pacific University in nearby Seattle, Washington. Many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music that makes me smile. I hope that you've all enjoyed being part of our quality quorum today. See you next time on Matters Microbial.